How has our understanding of Fragile X syndrome changed over the last 20 years? Dr. Mark Baer of MIT joined the Fragile X field in 1999 when he received a research grant from FRAXA Research Foundation. At the time, we recognized the symptoms of Fragile X, and we knew its cause, a single missing protein, but we knew very little else. Dr. Baer traces the discoveries that now give us great optimism of finding effective treatments and ultimately a cure for Fragile X.
Well, when I joined the field in the late 90s, I would say the situation was that the gene had been identified, some of the functions of the protein had been discerned, an animal model was available which was showing some changes in neuronal structure and also in the status of inhibition in the brain. And so that was sort of the initial state at the time. With respect to developing medicines, I don't think there was much hope that a medical treatment could be devised that would correct core pathophysiology of the disease. Rather, we were confronted with symptoms of the disease like irritability and anxiety that could be potentially controlled or ameliorated in a palliative way. I think the picture changed quite dramatically early in the noughts, 2002 and onward, with the discovery that many of the features of Fragile X syndrome could be accounted for by altered regulation of protein synthesis in neurons. That was one side of the coin. The other side of the coin was that there were specific neurotransmitter receptors and signaling mechanisms that control neuronal protein synthesis in a significant way. And so suddenly we had the prospect that we could develop a disease-modifying treatment based on restoring the normal balance of protein synthesis in the brains of those affected with Fragile X. This idea initially, of course, was wildly speculative, but it was quickly validated in animal studies, both using the mouse model of Fragile X and also zebrafish models and fruit fly models of Fragile X, showing an evolutionary conservation of this mechanism. So it was this insight that led to a lot of efforts in translating to the clinic, particularly developing the mGluR5 receptor inhibitors, mGluR5 being the glutamate receptor that triggers protein synthesis in neurons. As this story was progressing, we were beginning to fill in some of the other key functions of the FMRP protein. So it not only is a regulator of protein synthesis, but it also through direct protein-protein interactions can regulate the excitability of neurons through modulation of ion channels. And it's also, of course, been recognized for a long time that brain development in the absence of FMRP can alter the wiring of the brain such that the ratio of excitation to inhibition is altered, it's increased. So this gave a couple of different paths that one could take to develop disease-modifying therapies, one being specific modulation of ion channels that are affected by the loss of FMRP, and the other is more of a general approach of rebalancing this ratio of excitation to inhibition. So there's been a lot of progress over the last 20 years, and I think we have a lot of shots on goal.
This excerpt came from the Fragile X Syndrome in Pursuit of a Cure webinar that took place on World Fragile X Day 2021. It was hosted by Wuxi Aptech in partnership with FRAXA Research Foundation. Follow the link above or in the description below to view it in its entirety.